the technology is going to actually outpace our ability to understand what to do with the technology. Now we have whole genomic sequencing, which is going to give us an ungodly amount of information. And I think we need to decide when we get this information, we actually do understand how is that going to affect how we counsel patients.
It's the end of PGT as we know it. That's my title for this episode. My guest would describe this more as part of the great evolution of PGT. But what happens in evolution? The old ways die out. And so does any podcast episode title that isn't. So is this the end of next generation sequencing PGT? I invited these experts to tell me. Listen to their voices so you know who's who when they're speaking.
Hello, I'm Dr. Holly Mehr. I am at a practice based in Los Angeles. The name is Fertility and Surgical Associates of California.
Hi, I'm Dr. Arlene Morales. I'm a reproductive endocrinologist in San Diego. Our practice is Fertility Specialist Medical Group.
Hi, everyone. I'm Dr. Drew Tortoriello. I'm the medical director at Share Fertility Solutions in New York City.
Why do they think all PGT will include whole genome sequencing in 5 years? What are they afraid of with regard to PGT-P and direct-to-consumer market? What makes PGT-G different, and why are they guardedly optimistic about a lab called Juniper Genomics? What do they want to see from the ASRM and other societies with regard to a keynote speech on genomics? What do they want included in a large data set RCT? And why does Dr. Morales use Juniper Genomics for PGT-A even when she's not having the embryo's genome sequenced? These answers and more right now. If you like the information in this episode, we're compiling so much more data on new technology in IVF, products, services, market analyses, info on mergers and acquisitions, and data on every company in the fertility space. Go to data.insidereproductivehealth.com. If your company isn't an IVF Heroes Universe sponsor, your competitors are so much more visible to your customers because we report on your category and we give that info to your customers. Go to data.insidereproductivehealth.com to learn more about other fertility companies or submit your company today. Don't be left out.
Today's advertiser helped make the production and delivery of this episode possible for free to you. But the themes expressed by the guests do not necessarily reflect the views of Inside Reproductive Health, nor of the advertiser. The advertiser does not have editorial control over the content of this episode, and the guest's appearance is not an endorsement of the advertiser.
PGT was a dream 30 years ago. So PGT has evolved from the very early days of fish analysis, where it was just a goal to get chromosomes. Then we developed all this nomenclature of PGT-A for aneuploidy, PGT-SR for structural rearrangements, PGT-M for monogenic gene testing. And then over the past few years, we've been starting to hear of PGT-P, PGT-G. Again, what is all this advancement? And the advancement really is the technology. I mean, we're in the genomic boom, right? We're learning so much every year about genomics. And so I think the impression was that PGT, as we currently were doing it for the last 10 years, is probably going by the wayside eventually. And we're going to continue to advance PGT for further analysis. So we're going to start looking at whole genome sequencing. We're going to start looking at metabolomics. We're going to start looking at transcriptomes and RNA. There's a whole new field waiting for us to start identifying things in the embryos that we currently don't.
What are those specific methods or techniques or technologies from the last 10 years that you think will go by the wayside?
So currently, almost all of PGT-A is using next-gen sequencing. And over the past 2 years, what we've been seeing is that a lot of companies have started using two technologies. So they use next-gen sequencing, plus they're using an older technology, SNP arrays, or some other technology to make it stronger, the interpretation. One of the big concerns we currently have with PGT-A is this mosaicism. Is it a result of technology, or is it really a reflection of what's going on in the embryo? But now that the costs are coming down for whole genome sequencing, where you can actually start looking at the entire 3 billion base pairs, and you can start looking at RNA sequencing, we're starting to try to build a database of what that means in an embryo. I mean, there's so much data we know in adults, especially white Europeans, but we're learning more about what does it mean in embryo. We're just in its infancy. I mean, they're really, Drew and I were discussing earlier today that we're just building it. I mean, you can't open a Mendelian inheritance book and look at what does this mean in an embryo. We're just starting to learn what those variants that we see in whole genome sequence are. So the technology is really whole genome sequencing. That's a new technology.
Drew, Holly, is this consensus? Do you think that next-gen sequencing is going by the wayside, or are there people that still feel strongly about it?
I think that the next gen sequencing platforms are using similar technology. It's just that we now, with AI and other like bioinformatics capabilities, are able to get more specific about that sequencing that we're doing. Where with traditional PGT-A, they're looking at whole chromosomal components. Like, are these, 1,000 to 10,000 pieces of DNA present that we can identify as being part of chromosome 1, chromosome 2, chromosome 3, etc. And that allows us to see, is the whole chromosome there? But we're not utilizing the actual sequence of the letters that are DNA and RNA to ask ourselves, are those genes different in some way? Are they functional the way that we think they should be functional? And using new data mining, really, for lack of a better term, we're able to utilize that same similar technology. They're using different techniques, and that's a little bit outside of my realm of expertise, but it's only been in the last few years that they've really been able to reliably get that information from the sequencing that they're doing. And that's been the major change, is that we have some ability to believe that the data that we're getting, that the sequences that they're running, the actual letters of the DNA, are representative of the DNA that's been biopsied. Because when you think about what are we doing in any genetic testing that we do for anybody, we have to take a sample of DNA and then amplify it so that we can get some sort of signal, right? We replicate it while we're doing the testing. And when you are talking about an embryo that's only 100 cells, the amount of DNA that you're getting from that biopsy is so tiny compared to even just like a cheek swab or spitting in a tube, that our ability to believe that amplification and replication is real, and there aren't a whole bunch of errors in there just from the machines doing the amplification. And that's what's changed. And now all of a sudden, because we have more ability to believe all those letters that are there and all those sequences that are there, that amount of data is like crazy huge, right? It's like trillions, I think, of base pairs. I don't know, but
3 billion base pairs. Yeah, 3 billion.
But still, that's more, like, you can't even conceive of how large that number is, right? And so you can't really rely on like regular people, humans, to like look at that data and be able to understand where there's errors. You have to be able to compare it to something using computer models, etc. And so I think that's where the major shift is right now with the companies that are starting to do whole genome sequencing. It's just a little bit nerve-wracking that we're like putting all of our eggs, as it were, into the basket of we're going to believe the data that's coming from 5 cells of information, and so that's, I think, where I don't think NG, like the regular next-gen sequencing, looking for the embryos, chromosomes being there, is ever going away. I think just there's going to be a lot more opportunity to be looking for something much more specific for people, if that's what makes sense for them.
I think the technology is going to actually outpace our ability to understand what to do with the technology. When DNA arrays first came out, prior to that, we're doing northern blots and western blots, and now we have this incredible boatload of comparative DNA sequences, and then we're trying to suss out a disease connection between certain genes found on the array. Now we have whole genomic sequencing, which is going to give us an ungodly amount of information. And we're basically now, as Holly was saying, trying our best to keep pace with this information. I don't think it's any coincidence that AI and whole genomic sequencing are coming together as a synergistic duo, because you have incredibly potent information. Now, finally, with the mechanisms by which you can analyze that information, I think we need to decide when we get this information, we actually do understand these certain polygenic risk scores that we get for particular complex common diseases. How is that going to affect how we counsel patients? That's my mind goes to ultimately how we're going to be able to talk to patients. And I think about how we were talking to patients about mosaicism in the embryos that we were doing just regular PGT. Firstly, in New York, we weren't even allowed to know about mosaicism. You'd actually got to call the company and say, I'd like to specially get this information, please. And now we get the information fairly routinely, but for years we didn't get it. For years we didn't know how to approach it. Was it something that we could work with or not? And now finally we're just wrapping our arms around it. So I do think the technology is going to go all the way, whole genomic sequencing, and everything else is going to go by the wayside. I'm curious to see how AI and typical Janes and Joes like us talking right now actually get to explain that to our patients one day. That's going to be the crux, I think. It's going to be a team probably of geneticists, physicians, and medical ethicists.
Yeah, I 100% agree with you. Like that's the real concerning barrier is, the people that are sitting on this podcast right now have, and my husband points this out to me, he's not in medicine. And when I'm talking about my understanding of things with him, he's like, you are like, you feel as if this information and knowledge is accessible to other people, but you forget that it took you 12 years of training after graduating from college to like feel comfortable having this conversation. And even for me, I'm struggling. I just had a consult, a couple days ago with a patient where I was looking at their genetics report and just going, oh, geez, I'm not sure I can have a conversation about the complexity of this with this patient. And so I think the way that this is being marketed to the general public is very not specific, it depends on the platform or whatever, but just like generally the way the general public is understanding what this new technology means is very far off from what we can actually provide at this stage with, PGT-P, for example, as one example with polygenic risk scores. And also like when is it our job as the physician to break this all down? And when should we be passing that off to a genetic counselor? And how do we then know as the physician that we feel comfortable that the patient is coming away with an understanding that is appropriate? Because there's a lot of gray area in a bunch of this testing that's not black and white, especially as we're just trying to gather information with, variants that are less likely to lead to a live birth, et cetera. We're really in the information gathering stage right now. And so I think that part is really challenging. I totally agree with you, Drew. That's my biggest concern moving forward.
I think you did a great job stating again, the next gen sequencing, that's not new. What's new, and I want to be really clear about this, what's new is the bioinformatics and the ability to understand the 3 billion letters that we're getting. And it's only in January of 22 and August of 23, which is like a minute ago, that the first time the entire human gapless genome was published. Back 15 years ago was the 95% of it was done, but the entire sequence of non-coding just came out in August of 23 for XY. So that's really less than 3 years ago. So we're in our infancy in adult genomics and in what we know. And when you're saying the difference between PGT-P and PGT-G, to me, it's completely different. PGT-G is actually looking at the genome sequencing in single cells. I mean, you can do that in adults for a long time. What it means, I agree with you completely. We don't know what some of these variants mean. And so that database has to be built eventually. So what does it mean to have a variant? We don't have a Mendelian book that you can look it up and say, okay, this means you're going to get this disease or this thing. We're building that in embryonics, okay? But PGT-P, just to be really clear, is a statistical association. There's nothing to do with gene reading. It's a statistical association. And probably the most important thing, if you're going to remember, genome-wide associations in adults, mainly European males, pretty good data there. Associations with cardiovascular, diabetes, statistics, okay? But gene-wide association in a neonate is different than an adult. Even monozygote twins with type 1 diabetes, it's in Europeans maybe 60, 70%, but there's still 30% that's not. So these are statistical associations. So to assume in an embryo, you're going to say it has a statistical association with an adult, to me is outrageous. It really is. I mean, we're so in the infancy of learning this. That's very different than saying there's a genome, there's a specific variant that we're seeing. So very, very different. PGT-P, which is a statistical association. And you're not even taking into account, if you guys remember, genome-wide association, there's epigenomic-wide associations that we're just starting to look at the methylation patterns of people. I mean, that's now in adult genomics, you're looking at the combination of genome-wide sequencing and epigenetics. So we're so far away from that in embryos that I don't see, hopefully we get there one day, but we're far from, if you're marketing, we're talking about looking at single gene, actual reading the sequencing and doing the bioinformatics on that.
I think that beckons to Dr. Mehr's point that the general population not only do they not know the difference between PGT-P and PGT-G, the general population, they don't know the difference between genome sequencing versus genome editing versus CRISPR. All of this is in one black box to the general population. The general fertility patient is usually a bit more self-educated and is probably at the point where they're maybe learning about the differences between PGT-P and PGT-G, but it's probably still, they just kind of mix those two things together. And many of them maybe even don't even understand the difference between that and PGT-G, PGT-A. So, Drew, how does that affect, as you're talking about, we need to learn how to counsel these different ways. How are you counseling on these different things now?
For me, it's an evolutionary process right now because I haven't really partaken in this technology all that much at this stage. So I find I'm very humble about it because I have patients that come to me that actually in their desperation have been looking for any modality possible to try to give them an understanding as to why they're not succeeding. Half of what I know about, PGT-P and PGT-G comes from patients being the impetus for me to know about it, because they're very much interested in it. That's actually how I developed an association with Juniper, because I had a patient who came to me and said, I've had nothing but failures. I'm a young woman making what should be quality embryos, never get anywhere, never make a blastocyst. And it was under her impetus to actually try to do something with Juniper. And they found an association with something that seemingly has a physiological relationship with what might be going on with her embryos. And that gave her some closure. It gave her an understanding that there may be something which is somewhat insurmountable, and no protocol on God's green earth is going to give me a quality embryo. So how do I counsel them? I counsel them that it's a nascent technology that has tremendous promise. I don't recommend it for everybody because I think the data is very unwieldy and is way more than most people need. But for people that I think need some sort of genuine further understanding and really want to go to the final limits, it's a technology that can give some potential clarity or some potential direction.
And so how did you find this lab, Juniper? What made you go with them as you're learning about these different technologies and labs?
I think I was her fifth physician after so many IVF cycles that went literally nowhere. She said, look, she's on all of the Reddits and the chat rooms and this, and said, I've come across in one of my chat rooms or whatever about Juniper. Apparently they offer this technology which tells you more about your embryos. I want to know literally every last little bit of information I can get about my embryos. I told her that this likely is not going to change her prognosis, but if she's looking for some form of additional information, this seems to offer it. And in her case, I think it actually was paid dirt because she got a bit of information that made her understand that she has to go a different way. And actually she did. She went thereafter to donor egg IVF.
Arlene, how do you explain the difference to your colleagues that maybe still see PGT-G and PGT-P as very similar?
I think if I had to use one word, is one's in a statistical association and one's reading 3 billion letters of oneself. I mean, it's very, very different. One is literally a mathematical model. literally. And if you know that genome-wide associations are different in neonates and adults, how can you possibly then think that there's nothing? I mean, it has to be an evolution. And with genome associations, the epigenetic influences on top of these statistical uh, variants has to be influenced, right? So in an embryo, who knows what the methylation patterns are? I mean, I'm sure there's some research. I'm unaware of that. Again, I come to this field very humble. We are not trained as genetic counselors. Although I certainly feel as a reproductive endocrinologist, I'm a team leader, and part of that team is learning about genomics, about the advancements of what's happening. And so, as we grew up with this field in the last 30 years, our understanding of genomics has blossomed. Like I'm saying, we're nowhere near the end of this. I live in San Diego, where we're one of the genomic capitals of the world. I have people who think genes are what you wear genes, and then I have people who have been sequenced, and it's all the same patients. There's such a wide variation. It's very, very different the people we see. So it really depends. I'm certainly not at all recommending even PGTs for everybody. I think when we see patients, what I still love about what we see the 20-year-old who literally is anovulatory and needs ultrasound to conceive. We see the 42-year-old who wants 5 children who just got married. We see all kinds of patients that present. It is a privilege, I'm sure all three of us believe, to work with all these different people, couples who come to us to try to have families. And the first question I always ask someone is, what is the reproductive goal? Why are you presenting to us? What is your goal? Is your goal obviously to have a baby, but in what context? How many, over what time period? And so I certainly don't believe that PGT is maybe appropriate for everyone today even, but I do think that the advances continue. And I think we'll look back as this is one of the big advances.
So when you all say that you think that everything's moving towards whole genome sequencing, do you mean you think that once the technology, scale, and cost allow, that all PGT will be whole genome sequencing? Am I understanding that correctly? Will there be use cases once those things are true for PGT that doesn't do whole genome sequencing?
I guess with PGT-A, you're looking at the top of the mountain, right? You're not looking at the whole mountain. And the technology is the same. So if the cost is a non-issue, and now you can read the entire 3 billion letters, read just the top of the book.
Holly or Drew, do you see it that way as well? Or is there a reason just to read the top of the book?
I think as costs come down and the amount of data you can get cheaper, I think it's inevitable that they're going to wind up getting all of the genome sequenced for everybody that wants to do this. May not happen overnight, but I think within the next 5 years, probably.
I think it brings up some concerns with medical ethics and with, again, patient counseling, right? Like having all of this information and getting them to understand this embryo is found to have a variant that doesn't exist, that we haven't seen sequenced in a living human before, right? So that is what part of what Juniper is doing, is they sequence genes and see these like variants that seemingly are not compatible with life, but because we don't have the data that this variant has ever been seen in a live person, I think getting to the point where we can confidently say that that is true, that that is a fact, rather than this association that we found in the data that we have, is going to take time. And it's also, I think, I would really like there to be, in the same way that many clinics came together to provide their mosaic transfer data in the last 5 years or whatever however long we've been doing that study, it would be really helpful for there to be a big data set that we're all collectively using to help build these. And I don't know enough about the industry. I don't know enough about how these scientists that are venture capital backed scientists that are, they would, I think, probably would like a lot of their data to be private and proprietary. I'm not working there, but that is, I think we need a lot more information before we can start feeling confident about what we're telling patients, and how we can counsel them about the like gray area that we're in right now is a little bit challenging. Like certainly for somebody like Drew's patient, who she's done so much, and to like find an answer finally, something that makes sense logically for her, is a really great place for her to be. But what if that person, they had only done one prior IVF cycle, and they came and had that same outcome, are you then going to tell them that they shouldn't be continuing to try and get pregnant with their own eggs and they should move to egg donor? I don't think anyone, I don't know, I've yet to hear the argument that we understand what's going on definitively enough that we can make that educational recommendation or that recommendation to clinically to a patient. Personally, I feel super hopeful about it, but I just think I think Juniper's only been around for like 6 months or something. And so they're gathering information from really reliable sources of probable genetic difference in our population from like these very sort of extreme cases at this point, I think are often the folks that are ending up at their door. But in 5 minutes, I can tell you that there's going to be a whole bunch of people in Los Angeles, in like large metropolitan areas, who are going to also want just the best. They want the best. They want to know everything there is to know. And I don't know that we're ready for that yet. Like this, I feel like my own personal opinion is that at this moment, this technology should be for the people who really are looking for an answer that they haven't been able to get in all the like sort of traditional ways we've looked at this before.
Arlene, what further data do you want to see? And for what use cases do you think there is currently sufficient data?
I think there's a tremendous amount of data that we still need before we can certainly recommend this technology for everybody. I think Juniper, the reason that attracted me to that company specifically is the initial founders were also bioethicists and molecular biologists. Bioethics is a huge bioethicist is a huge part of this, right? How to apply this technology. I think reproductive endocrinologists truly are at the nexus of genomics, technology, and bioethics. I mean, you said earlier, Holly, if not us, who? I mean, we are the leaders in this field. We are the ones, the attorneys or anyone else gets involved or the VCs. We are the professionals that are supposed to be at this nexus of in reproduction. So I think even when you get genome sequenced yourself, if any of the three of us went to anywhere and decided to get sequenced, it's not that easy. There's about a third difference depending on what company you decide to go get sequenced. There's a lot that we still don't know. So one of the nice things about Juniper is they'll still do PGT-A. That's the initial goal is PGT-A. And then if you need to, they've already run the 3 billion base pairs. The bioinformatics hasn't been run. So if you have to go backwards and see, okay, why didn't this go? You can go backwards. The data's run there. They haven't done the bioinformatics, but the data's run. So that's one of the main reasons a lot of times I'm using right now for PGT-A is we still are just looking at the top of the mountain. We're still just looking at the 23 big chromosomes, the major reason for miscarriages we all know based on cytogenetics. But it gives you the ability to deep dive if you need it. And not everyone needs it. I couldn't agree with my colleagues more. And we're just starting to figure out who needs it and building databases about what do we see in humans, what do we have not seen in humans. But I'm certainly not thinking that PGT-A and the way we do it currently is dead. I think that we're evolving, I think is the term, as we continue to evolve.
And that's why a lot of people use Juniper, right, Arlene, because they use it for PGT-A, and then if they want to sequence, they're already with that lab. So
the information's there. The bioinformatics hasn't happened. That the actual technology has happened. The sequencing has been done. They just aren't looking at that. The curation of that data is what's really is difficult and costly. Not necessarily the sequencing by itself, but the interpretation and the bioinformatics and using world databases, the European, the American databases to see what variants are important and what are not. And those are human calls, right? And so I think you said earlier, we don't know for sure that variant will always do that. And that's what we're learning.
Yeah, Holly, when you ask the question, if not us, who, the answer is anybody who likes money is who's going to try to fill that void. And the rest of us that don't have advanced degrees or people like me that have humanities degrees from some state school that the rest of us never heard of, we're left to try to figure it out for ourselves and say, well, maybe this fertility specialist knows what they're talking about, or maybe this other person that tells me is an expert is equally an expert because they're telling me that they're as much of an expert as this reproductive endocrinologist, and I don't have a way of comparing their credentials. And I'm thinking of at a recent conference, someone came to me from a PGT-P lab and showed me a deck and made claims of that, no, we can show, we can influence height, that we have the evidence to prove it. I don't have a way of refuting that. And so how do you, as an REI community, be the ones to take charge and I guess maybe you lay out the requirements that you need to see before any kind of these solutions go to further scale?
I agree with you. I think that is correct. I think as a professional group, I don't know, at least I haven't seen at our conferences as much of a push for that as I'd like to see. Like in reflecting on this topic, I thought, I haven't seen, I would really appreciate if there was like a keynote address at our national conference specifically about this, so that I think it's, as practicing providers, we have to work really hard to like stay up on the new technology that's available. And there are companies that are going to have direct consumer marketing already for technologies that most physicians, I think, don't have enough like comfort with to really be able to critically evaluate. And I've had most of the like continuing medical education I've gotten around these technologies have come from the companies themselves reaching out to try and provide more information, which I think is very valuable. But they clearly have, and without even meaning to, they have a bias towards projecting their data as something that's valuable. And we've gone through as a field many, many, many iterations of this. We are working in a data hole, as you know, in the women's health field for reproduction. We have so much less research funding than most other medical fields do. Plus, we're technology driven. And as humans, we tend to be people that, like the physicians who have gone into this field, tend to be willing to be early adopters of technology because of what REI is. And we're in the United States of America in particular has like really been pushing the needle when it comes to a lot of IVF changes, with whether it's ICSI or PGT-A or next gen sequencing or whatever. So in the United States of America, we are sort of in this, there's not shared data that we're all using. There's no national registry. I mean, there is, we have our SART data, but that's not the same thing as what other countries are able to do. And so I think it's really imperative, like this feels like a sort of tipping point is happening, to me at least. Another one where, we were thinking about the end of, we've been sitting here hemming and hawing about why is our pregnancy rate stuck at 55% for embryo? And we've all been asking the question over and over and over again, is it the embryo or is it the environment? Is there something that we missed? Should we be testing something inside the uterus? Is it the ERA? Is it receptiva? Is it a biopsy? Is it a hysteroscopy? Should we be putting them on Lupron? Should we just have all of these questions about like how to improve pregnancy rates when it seems like the data is pointing to the answer is in the embryo, but we're sort of relying on these outside companies to like mine that data for us and like create the platforms. And I'm very glad that it is happening. I think it's really beneficial, and I would love to have, Juniper is saying some of their new data is like an 80% pregnancy rate per transfer. That's amazing. But I would like to make sure, I don't know how to do it. I feel like part of the issue is that like less than 1% of national funding for research is going to our field. And we're relying on venture capital backed companies to do the research for us. And it just makes me concerned that there's going to be this conflict of interest in what comes out of it. Again, this is not the first time that we've been, there was the whole question of mosaicism, the question of what to do with an endometrium. Like we've had these problems over and over again where we like get new technology and new information and misinterpret what it means because of somebody's interpretation of the data. So I'm very excited about it. I think it's going to be changing, very changing for our field. I just worry about adapting to it or adopting it too early.
Who would give that keynote? Would it be an REI or would it be a geneticist?
I don't know. It's a very good question. A geneticist, probably. But it's like somebody who's currently retired. I don't know. Somebody who's not working on behalf of one of these very important companies.
They're on the pay more than anybody else, the retired folks.
But as a society, I mean, we can look at different countries, right? I mean, the United States, autonomy reigns supreme, right? I mean, that's part of the issue, right? You saw in New York City, the buses and, when people are saying advertisements and stuff. But, you look at the UK where when they want to bring in a new technology, they have an open forum for society for like a 2-year window for people to start talking about it and saying, hey, as a society, mitochondrial DNA, for example. But in the United States, we'll never have that, right? It's autonomy, autonomy, autonomy. And as physicians, I do think that we are the team leaders of that conversation with the patient. And you're right, we need better information gathering in our specialty. I'm sure, Griffin, you've had talks on VC backed, and in California are very strict. VCs aren't practitioners of medicine, and you can see that whole field of our. When I grew up, nobody had venture backed capitalists. The GIs went first, and the REIs, but that is changing. And I think when a patient goes to see someone, I think it's imperative if they understand what the goal of that clinic is. Right? Are they looking at maximizing pregnancy rates as an individual here? Are they looking at parental autonomy to be able to pick different things? I mean, there's all kinds of things within our field, but I couldn't agree with you more that we definitely need further education in genomics. And when people show you a slide deck, Griffin, I hope what we have that's a little different is we have feels like 100 years of physiological training of things that make sense to us. We shouldn't be hopefully BS'd by outside companies, because we should be able to understand, okay, make me understand it. Make me understand why this is true. And how can it be a statistical association when this isn't true? And so I do think that physicians do hold a special role, and that we do have a whole education of understanding science, right? And maybe you don't understand the actual field itself, but someone should be able to explain to us and should be able to critically think of saying, hey, what makes and doesn't make sense. So I definitely applaud more education in our subspecialty for genomics.
I'll also just say, Arlene, on top of that, this is not to like say ASRM is not doing good work. They are. I think, at the end of 2025, they had an entire special publication in fertility and sterility that was just about sort of laying out the expectations that we should be putting onto PGT companies for validating their platforms. And that was very, I think, useful to have as a resource. I think a lot of clinicians are busy and maybe not reading everything that comes across their desk, or not coming across their desk at all. So, some more accessible education, like podcasts, maybe, that the people of LA can listen to while they're in traffic, is also, warranted.
Do it for the good doctors in the high traffic metro areas. Drew, do you share this perspective that the topic of genomics is undercovered in the societies and conferences? And if so, why would that be the case? As you mentioned, you think it could be the case that in 5 years that all PGT includes whole genome sequencing. So why, if that's true, is this as undercovered as it has been?
I don't know if it's been undercovered per se. I think, it seems to me that most people, they get one minor finding and they're ready to shout, into the heavens about it for the most part. I think it may be under discussed with the people who need to know the most about it, which are the patients that are walking into our office. I really dislike this direct to consumer form of advertising. I don't like commercials on television for every drug under the sun, trying to get you to go to your doctor to get this drug and that drug. I feel similarly about the subway posters that are talking about have a child with a higher IQ, and they come to our office and start talking to us about it. So I think we are under the burden to try to make as much sense of this as possible, and then translate it to our patients about the relative priority for them and their fertility treatment. I find that most people think that it does things that it just doesn't do, and they want us to be an honest broker. It's hard to be the honest broker. It's hard to be the person that when somebody walks in at age 49 years old and heard about a technology that they're offering in Cyprus, that it may or not necessarily be the end all and be all of making a baby. It's hard to be the discouraging one from time to time, but sometimes we have to get it to what Arlene was saying. We have to be team leaders. Sometimes it's hard to be a team leader also when you are at the mercy of companies that are giving you their data, their information, in a selective and piecemeal and very advertising-like fashion, rather than as a true scientist or a true colleague might give it to you. So I think that for most people that are finding some scientific breakthrough, no matter how small, it does get out. I don't think there's a paucity of that information. I think the problem is what they do with that little bit of information to bombard the general population before properly educating the physician scientists that have to deal with the people that come to your office with it.
And also accumulating, really what I would like to see is it sort of referring back to ASRM's outline of how to validate a test. Like we really should be doing a form of randomized controlled trials before we are accepting the validation of any test. And that is pretty hard to do when we're talking about when the like randomization is an embryo, right? And so I think a lot of effort would need to go into doing that clinically. It would need to be a bunch of different clinics, a bunch of different doctors sort of signing up to do these randomized controlled trials, and a bunch of funding because patients are going to want some compensation for doing all this intervention without getting the benefit of knowing what the data is. And so I think that's maybe where we can like hold, I don't know, we could ask to start holding people accountable as like, I'd love to use you. When are you going to be doing your like blinded transfer study? Please let me know and I'll, pay for me to have somebody who's enrolling patients and pay for patients PGT with your company, and we'll get the blinded data so that you can be validated. That's just a thought. That's a big thought, but
Griffin, how do you validate for height using a technology that's 3 years old?
maybe best.
And to the people that are your colleagues, that's an elementary question. For those of us that are just seeing numbers and charts, it's the answer could be, oh, that's how they did it. And they don't know the answer to that, because in the absence of having a unified discussion, you will be bombarded by these requests from patients, and every REI will have to figure this out individually, figure out the response individually. Because if there's anything that I've learned in sales and marketing for the last 20 years, it's the American consumer gets what the American consumer wants, one way or the other. And so there needs to be a unified discussion from REIs and geneticists in order to be able to respond to that. Otherwise, it's going to be the venture capitalists and the financiers finding a way to just give the consumer what it is they want. And perhaps a randomized controlled trial is exactly what's needed. To your earlier point, Holly, getting that data is the hardest part in terms of big sets, because the big networks don't really want to part ways with their data. And if they do, they want a lot of money for it, because it's like, all right, you're trying to build this thing and you want us to give you the data, and then you're going to go off and make money for it elsewhere. It's hard to make those RCTs. But Arlene, if you were pitching to someone to participate in an RCT like that, what would be in it for them?
I think Holly and I are both somewhat unique in that we're transitioning in the, I should be careful how I finish that sentence. We're transitioning between an insurance mandate and not insurance mandated state in California. And to me, I don't know, I haven't asked you, but it's been starkly different. The patients that I've seen before December 31st of 25 and after 1/1 of 26. I mean, oh my gosh, very, very different.
What do you mean by that, Arlene?
So a lot of patients in California, with Senate Bill 729, was that any company in California, except for a few exceptions that has at least 100 employees, has to cover, well, first defined infertility is just the inability of a single person or couple not to be able to reproduce. And so it defined infertility for us as a medical diagnosis. That's the first. And then secondly, it said that if you're one of those companies, you have to cover 3 rounds of egg retrieval and unlimited transfers, a variety of other things in there. So we're really excited that we finally had some coverage for so many patients. That's been life-changing for a lot of patients. And our goal is still to extend that even further for other patients. But what's happened is that the financial piece is not part of the medical decision-making, but for patients, it's a big piece sometimes of what technology they choose. So I've seen patients in the beginning, it's been hard to sort of push back and saying, just because I have benefits, I'm going to use it. And I'm like, no, that's not the practice of medicine. Patients don't have a right to demand therapy. They have a right to decline therapy. It's very clearly written in the U.S. legislation that you can decline therapy, but you can't demand therapy that we don't think is medically appropriate. So a 49-year-old to come in and demand IVF because they have coverage, I'm not under any obligation to provide that therapy. And I think same with PGT and genetic testing. I think it's a real misconstruct that patients think they have a right to demand therapy. There's no physician has no obligation to provide therapy they don't think is medically appropriate. So it's a right to decline, not demand. And so I don't know, Holly, if you've seen that or not. But when it comes to genomic testing, again, I think I'm comfortable offering some genetic testing in certain circumstances, but not a one size fits all, I think, at this point.
Yeah. Yeah.
I don't know if you noticed a difference.
They have. It's been more that, I mean, I'm in Los Angeles, on the west side of Los Angeles. So I have a lot of patients who have coverage to their employer anyway, because there's a lot of large tech companies that are providing coverage through, these other ways of doing it, through these other companies. And so it's been different having a lot more patients actually of more, I guess I would say in my particular practice, of like less means that like this has allowed them to seek fertility care that they otherwise wouldn't have sought because of the cost for it, which is the whole point. Yeah. So it's very excellent. But it then again, a lot of, not always, but many times, those people who have less financial means also have a lower health literacy around any of this stuff. And so it brings in a whole nother level of complication when you're trying to talk about complicated topics like genetics, where our heads are spinning after, like basically swimming in it for our entire career. And so I think that part of it is a little bit more challenging, is like trying to strike the balance of like, how deep do I go down this rabbit hole with you? I don't want, like, I don't want to be staring at a deer in headlights across the desk from me, but I also feel like it's your right to have education around this. And so, I don't know, it's that part has been interesting for sure. And like, I'm still trying to figure out the balance of like reading the room, but again, people are showing up and just being like, yes, I have the coverage now. That means I want the thing. And you have to walk it back a little bit and say, this is amazing. I'm so happy for you. Let's talk about the way that this actually works. Like, first we do a workup, then we talk about your chance of about your prognosis. Like, are we doing something that is medically futile? Is this something that you don't need? And that has come up, I would say, for sure.
Drew, did you see more of that when New York switched to their mandate? I don't know if it was 5 years ago, something like that.
A bit of it. I've always had a patient population that's always been, I think, a little bit more, I would say, more difficult. Just that, we usually get people that have failed IVF cycles elsewhere and then come to us. So I'm used to getting people that will come in older. But to Holly's point, there's a tremendous number of people that the heart wants to help, but the mind says no. And it's a balance about how you deal with it. Sometimes, the physiology is just not there. And even the money might be there. We have a lot of people from abroad, actually. They don't care about money. They just want a chance. I just want a chance. Just give me a chance. Well, you're 49 or 50 and you've done 4 failed IVF cycles. It's not going to happen. I don't think I've seen the dramatic change that maybe the other 2 esteemed physicians have seen. It's always been, I think, more of an older population for me.
Didn't even get to ask about the change in counseling on mosaicism and how that consensus has shifted and where it's going. That'll have to wait for another conversation. And I look forward to having the 3 of you to go back to see how this topic continues to develop. Dr. Arlene Morales, Dr. Holly Mehr, Dr. Drew Tortoriello, thank you all for joining me on the Inside Reproductive Health podcast. Go to data.insidereproductivehealth.com. That's where we're compiling so much data on new technology in IVF, products and services, market analyses, info and mergers and acquisitions. Every data point that we can on the field writ large and the companies in the fertility field, we're compiling there. Go to data.insidereproductivehealth.com as we build that over time. And if your company isn't an IVF Heroes Universe sponsorship, your competitors are getting more visibility because we report on your category and we give that to your customers. We do that multiple times per year, and we do it around big events like ASRM and ESHRE.
Go to data.insidereproductivehealth.com and submit your company today. Don't be left out.
Today's advertiser helped make the production and delivery of this episode possible for free to you. But the themes expressed by the guests do not necessarily reflect the views of Inside Reproductive Health, nor of the advertiser. The advertiser does not have editorial control over the content of this episode, and the guest's appearance is not an endorsement of the advertiser. Thank you for listening to Inside Reproductive Health.